This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 549, recorded on May 24th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. This episode is sponsored by the 2019 ChemBio Defense Science and Technology Conference. Are you working on innovative research that can shape the future of chemical or biological defense? Submit your abstract and present your work to more than 1,500 leaders from government, academia, and industry. Visit cbdstconference.com for more details. cbdstconference.com. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it's pretty much a nice day here. We've got it's been some overcast rolling through, but it's 72 Fahrenheit and 22 Celsius and mildly breezy and just a nice late spring day.
It's pretty nice here, mostly cloudy, 23 Celsius. Yesterday was a killer. Lots of storms rolling through here.
Yeah.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's pretty nice here. I forgot to pull up the weather. I was daydreaming because the weather's so nice. Let's see. What does it say officially? 68 feels like 68. That's mean there's no wind, no rain. Yeah.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. It's 87 Fahrenheit, 31 C. We aren't going to see the 60s for a long time. Now we're headed towards summer, and it's I'm going to say, being optimistic, partly sunny, but that's probably over optimistic.
Good to have you all back again. I miss hearing your voices. It's been a couple of weeks. And we have, for your listening pleasure, we have two guests right here in studio. They are both from the Albert Einstein College of Medicine. One of them is returning for his second TWiV. He's also been on TWiVo. He's a professor in the Department of Microbiology and Immunology, Kartik Chandran. Welcome back.
Hey, Vincent. Happy to be here.
Good to see you again. And he has with him a research assistant professor, Rohit Jangra.
Hello, everyone.
Welcome. Rohit, right?
Yes.
Rohit Jangra.
Yes.
Is that how you would say it?
Yeah. So it's like Rohit rather than Rohit.
Yeah.
So
Rohit.
Yeah.
Just think about hitting and
yeah.
He doesn't hit you, does he?
No.
Rohit Jangra, is that the way to say the last name?
Yeah.
I've been accused of giving everything an Italian accent, So I'm sorry if I Italian accentize.
No, you are pronouncing it.
Rohit Jangra. Where are you from?
India.
What part?
Northern part. It's pretty close to Delhi, about 4 hours
Delhi.
drive from them. From there, and I'm from countryside. So my village name is Dhansu, which in Hindi actually means tremendous. So
tremendous.
Yeah.
That's tremendous.
Nice.
And how far did you go in your schooling in India?
So I did my high up to 10th standard, because in India schooling system is a little bit different. So up to 10th we call it high school, and then 11th, 12th is sort of like college. So up to 10th I was in my village itself. Then I went about 10 kilometers away to the nearby town. And then I went to vet school, actually.
In India?
Yeah. So I trained as a veterinarian. Somewhere in the, about third year of my vet school, I was like, I don't want to do clinic. I should look into doing research. But all the inspiration, actually, to be in vet school and to be actually doing science it came from a lot of people, especially, my uncle, my cousin brother, who was actually the first one to do that. And in village of about 10,000 people. We were among the first ones to actually go to college and study science. And that was pretty interesting because my parents, they never went to school even. So
You're a pioneer.
Well, I wouldn't say that. I would call my cousin brother as pioneer. I just followed him and his guidance.
Cool.
Yeah.
So did you finish vet school?
I finished vet school, and then I was like, how do I get into research?
Right.
So in India, there is this premier institute for veterinary research called Indian Veterinary Research Institute. So
that's easy to remember.
Yeah, right. Back in the British days, it used to be called like Imperial Veterinary Research Institute.
Of course.
So there, I liked viruses, so I figured I would do masters in veterinary virology. So there, I studied a picornavirus, foot and mouth disease virus, looking at how the virus changes. So it's pretty endemic in India. Even now, even though there is a vaccine, there are certain areas that have been declared free of FMD, but still it's a quite significant problem. So it's looking at antigenic and genetic variation in the field isolates and compare that to the vaccine and how the vaccine wasn't providing good enough coverage.
What did you do after that?
So after that, in 2004, I moved to UTMB at Galveston for my PhD, where I worked with Stan Lemon.
Yeah.
And actually worked on role of microRNA, the liver-specific microRNA, MIR122.
Hep C. Right?
Yeah, and its role in Hep C replication. Actually, some of those papers were mentioned way back on TWiV that I worked on. So I was really happy to be in Stan's lab and to do some interesting work there and follow my interest in virology, especially working on microRNAs. So graduated from there in 2010. I was at Mount Sinai for a couple of years in Benton Hoover's lab, working on microRNAs and influenza. And then I was looking for a change. So Kartik was looking for a position at that time. I'm like, I should do something different. And of course, still viruses, but started working on virus entry. So moved to Kartik's lab in June 2012. And I've been there since. Yeah.
The rest is history, as they say.
Yeah, exactly.
And in today's paper, you're the first author, or co-first author, so we're going to talk about that. Ben was just here last week, gave a talk.
Oh, nice. Ben
Hoover. I was trying to find his papers, and I forgot you're not supposed to separate the 10 Hoover.
Yeah.
They're all one.
Yeah, it's one word.
If you separate them, you get 2 papers on PubMed. And I said, no, he has to have more than 2 papers. Isn't that funny how that happens?
Yeah.
Kartik, remind us of your history. I forgot.
Yeah, so I also grew up in India, but I think I have a background that's about as different as you can get from Rohit's growing up in India. So I grew up in South India, in a major metropolitan area, Bangalore, and finished my schooling, finished high school there. And then I actually, wanted to be a biochemist, and I didn't want to, I don't know. This is like the late 80s, and, everybody wanted you to be an engineer or something, and I wasn't interested. So I just kind of on a whim, I applied for colleges in the U.S., and I was, incredibly lucky that I got a full scholarship at Lafayette College in Pennsylvania. So I came out from undergrad, got a bachelor's degree, and then I did my PhD at UW-Madison, Wisconsin, and then moved with my PhD advisor, Max Nybert, who's working on reoviruses, and that's what I did my PhD on, reovirus entry. And so he was recruited to Harvard Medical School, so I came out with him as a senior grad student, finished my PhD, stuck around for a couple of years, and then worked with Jim Cunningham at the Brigham. It was a retrovirus lab, but I kind of, 2 months in, I was like, I want to do something else. And Jim was great. He's like, do whatever you want. And so I decided I would use the retrovirus system to pseudotype Ebola glycoprotein, which Paul Bates had just published a paper on that, and Yoshikawa Oka. And it looked like there were a lot of interesting questions. And then very quickly I just started to use the VSV system because Sean Whalen was there, and he's like, just do it in VSV. So, and then we made some interesting observations on Ebola entry mechanism, which allowed me to get a faculty position at Einstein, moved there in 2007, and I've been there ever since.
Okay. So some of that I remember. Yeah. The Max Niebert part I remember. Yeah. R.I.P. Michael Rossmann.
Yeah.
Too bad. 88.
88.
Yeah.
Died with his boots on. Mm-hmm. Never left the lab.
No, he didn't. Kept working to the end. Now, what happens to his lab is an issue, of course, but everyone's going to have to find other positions. Well, over the years, we have talked of many of his structures solved in his lab. And in fact, back in I don't remember the year, a couple of years ago, I interviewed Michael for the Principles of Virology. And so I will put a link to that interview, which is really good. And he talks about his early days in Germany, moving to the UK, and then finally to the US. I remember at the end, I asked him, computing has changed so much. I mean, he talked about how it revolutionized structural biology, right? The power of computing. I said, what do you use now? He pulled his phone out. He said, this. Yeah, good interview.
Michael was really a structural biologist par excellence, and I think of him as basically a pioneer in doing structures of viruses. So many of the viral structures that we have come from his lab, and most recently, a lot of enormous viral structures, and an extraordinarily enthusiastic scientist who was just a locomotive.
Yep.
Amazing.
And I have to mention, because I'm an alumna, he's at Purdue University in the Department of Biological Sciences.
Indeed. Purdue University. All right. Let's talk about a paper that, it has 3,000 authors.
Yeah.
But there are four co-first authors. And Rohit, you got the first position. Jay,
first author.
The first author, which is always nice because, for obvious reasons. Protocadherin 1 is essential for cell entry by New World hantaviruses. This came out last year, right?
Yeah.
November. And so we've been meaning to get you guys out here to talk about this for a while, so here we are. And
so the other first authors are Andrew Herbert, Rong Li, and Lucas J? Or is Jai?
J.
Okay. And Kartik is the senior author, and then there are a bazillion people in between them.
There's actually multiple senior authors as well.
I'm sorry.
4 of us. So there's a symmetry at least, right? There's
yes.
4 first and 4 senior, and that also...
Romacap, Wang, Dai, and Chandran are the senior authors, I see.
So Felix Ray is on this too.
Yeah.
What did he do?
So we are collaborating with Felix's group on solving the structure of this protein. Although there is no structure, and they're still working on that, but they actually did a sort of more biophysically rigorous binding experiment of the virus and the receptor in this paper, which we had, that was something that the reviewers wanted, and we had tried and struggled a little bit to get that done. And so I reached out to Felix and said, hey, you guys want to give it a shot? And he's like, sure. And he had 2 postdocs in his lab who are both authors on the paper, Gleda Roman Sosa and Pablo Guardado Calvo. They were able to get that assay to work, and so we put that data in the paper, and we're happy to include them.
We always speculate on TWiV which experiments were asked for by the reviewers.
Right.
Happy to tell you.
And now we know, for this paper at least. All right. So let's go through the science here, but let's start by talking a little bit about hantaviruses. Tell us what you know.
Yeah, so hantaviruses are this pretty interesting group of viruses that are, some people say that these are the most prevalent zoonotic viruses that's going to even become bigger because these are carried by, at least they can be carried by different species, but the viruses that cause disease in people so far are largely associated with rodents. And given, the changes that are happening in climate and everything else and encroachment of habitat, we are likely going to see more and more of, rodents coming in close proximity with people and likely see an increase in these infections. So largely, just like arenaviruses, you can divide hantaviruses into, 2 groups, old world and new world viruses. and they seem to associate with two different diseases. In the New World, they cause what is called hantavirus pulmonary syndrome, which causes more of a respiratory symptom. Whereas in the Old World, it is associated with renal disease. So there are kidney impairment, kidney function impairment.
These old worlds are also rodent transmitted, right?
Yes. All of them are carried by rodents, but they are associated with different species.
And what impact do these have on the rodents?
As far as we know, none.
Well, they cause persistent infection in the rodents. They seem to be carried essentially for life. And there's a lot of evidence. Rohit was just saying. So just the basic virology. These used to be considered bunyaviruses. I mean, they are bunyaviruses, but now they are in their own family, hantaviridae. And these are negative strand RNA viruses with segmented genomes, enveloped particles. It's quite striking that the distribution of these viruses, which is worldwide, and different kinds of hosts, not just rodents, but also moles and shrews and bats. And there have even been, hantaviruses found in fish. So there's this is one of my favorite fish names, mini pizza batfish in the South China Sea that has a that's infected by hantavirus. And so one thing you see with these viruses is that they seem to co-evolve with their hosts. And so there's a notion that the viruses that we're isolating from these different animals have co-evolved with their hosts for long periods of time. But there's also some, genetic evidence for virus to jump from, one kind of host to another and sort of, take up residence, so to speak, in this new host. Because you can see these patterns of sort of alignment or symmetry between evolution of the hosts and the evolution of the viruses, but you also see viruses that are sort of these cross-down viruses, if you know what I mean. They're cutting across all these lines of coevolution. So, I think it's a fascinating family of viruses, and we know very little about them beyond having sequences and, a lot of these more exotic viruses. For, I guess, maybe reasons that we understand, that there's been a lot of more effort on the human disease-causing viruses, but this is a much larger family of viruses about which we know very little.
Right, and the ones that jump into humans are mostly in rodents, as far
as we know. The only viruses, well, actually, Thotapalium is a shrew-borne hantavirus, and actually was the first hantavirus, right? Ironically, so yeah, the major human disease-causing hantaviruses appear to be rodent-borne, but it wouldn't be surprising, I think, to any of us if viruses from other groups could also cause human disease. We just don't know.
So is it correct that C. nombre was the first discovery of the New World hantaviruses,
yes,
in the Four Corners region of the U.S., right?
Yeah, so that was the discovery in early 90s, 1992, 93.
Yeah, after that.
So
of hantavirus pulmonary syndrome, right?
Yes.
But we had already known about the Old World.
Yes, exactly. So the Old World one, in terms of disease, it was known from the Korean War. So there were many U.S. Army personnel who were falling sick, and not only U.S. Army, but I think Japanese Army and Russian Army was also having similar issues. But the virus was not isolated. So that was 1953. The virus isolation happened in 1976.
My year of birth, 1953.
And so
not 76. No.
Yeah, the first virus was discovered as the hantan. It's called the hantan virus,
right?
Which is sort of a geographical name.
South Korea, right?
Yeah, and that the type virus, I guess, gave the family its name hantavirus.
I think there's a Hantan River.
Yeah, Hantan River. That's right.
So it's funny, shrews. So on my TWiV from Rotterdam, Martin Beer works on bornaviruses in shrews. And he said, as far as we know, I can't do his accent, but as far as we know, the shrews are okay, but this, what do we know about shrews?
Yeah.
Right.
That was a funny line.
As far as we can tell from a shrew. It's funny because we always say,
just to clarify, it's the same thing in the rodents, right? It's a persistent infection, but apparently asymptomatic. Is that correct?
Right. And there is little evidence of any pathology. So there even has been some experimental studies so deer mouse is actually the reservoir for C. nombre virus. People have done some studies. I think Tony Shauns was involved in some of those studies where putting Cynombre virus in deer mice and looking at pathology, there is little evidence of any pathology in different tissues. Yeah. So they seem to be fine.
They just have this virus and they shed it in their urine, if I recall correctly, right?
Yes, urine and feces, and usually people get it when they are, trying to do their spring cleaning.
Yeah, right. Aerosolize,
And
sweeping,
the animals,
sweeping the mouse poop out of the shed.
So the rodents could give it to each other by biting as well, right?
Yes.
Okay.
And in the U.S., are there different strains of hantavirus that co-evolve with different strains of mice?
Yes. So actually there are, so some of the other viruses that have been associated with the pulmonary syndrome in U.S. include New York 1 virus, which is in this area. I believe the rodent is different from the deer mouse. And then there is a Black Creek Canal virus, which is in the southern
I love that name.
US.
Yeah, it's
love that name.
I think it was found and first discovered in Louisiana. And
near the Black Creek Canal, I'm going to guess.
You got it.
So there are multiple viruses and multiple species involved, and they seem to be again associated with like a particular reservoir rodent.
Paramiscus maniculatus.
Yes, that's the host for deer mice.
So I spoke with Jason Botten. Does that name ring a bell? He, up in Vermont, TWiV 240, virology in Vermont.
Jason is a fellow hantavirus enthusiast.
And I have a picture of him. He studies these wild mice. He said if you catch mice, about 30% of them have hantaviruses in them. And he takes 55-gallon drums and he sinks them into the ground up to the rim and then puts the mice in them. So they're really outside, but they can't get out.
Right.
And I said, who does the sinking? He said, I do. I dig the holes. And he's got a picture with 1, 2, 3, 4, 5, 6 of these things.
So that's like a silo for rodents or something.
Yeah, he was the one I think he was the one who did it for the first time as part of his, I don't know if it was postdoc or PhD program. And
he'd come from New Mexico or somewhere out there. Right, right. Now, this is a new virus for your lab, right?
Yeah.
Yes.
So where came the idea to do this?
Actually, around the time I was about to start in the lab, Kartik was thinking of moving beyond filoviruses. So when I was interviewing, he was like, well, we are interested in these hantaviruses. And I was like, I know nothing about them, but let's find something out. So one of the lab mottos that we have is like, we want to study any virus, we just say like, put it in the VSV. So basically take the glycoprotein of any virus that we want to study. Of course, it doesn't work for every virus, but it works for many enveloped viruses. So we started by putting the glycoprotein of one of the New World hantavirus, Andes virus. Actually, we haven't talked about that yet. It's the one that is present in South America and many countries.
In the Andes.
Yes.
In fact, there's an outbreak.
I'm good at this.
There's an outbreak going on. It's been sort of a low intensity outbreak in Patagonia for some time now. That's still ongoing with this virus.
These are BSL-3 viruses, right?
Yes.
Okay. And do you guys have a BSL-3 at Einstein?
We could. So, we do have BSL-3.
Depends who's asking, right?
Well, I think, the issues are more logistical for us. We haven't done any BSL-3 work with these agents. We tend to like to do BSL-2 work because it's a lot easier or faster. And BSL-3 space is limited at Einstein. But we also have great collaborators who are willing to start working on things because we want to work on them. So, for example, the other folks on this paper, John Dye, John's at USAMRIID, Fort Detrick outside of DC, and they, John's an immunologist by training, but really sort of started working on Ebola and then collaborated with us. And then, at some point I was just like, hey, we want to start working on hantas. Are you guys interested? And he was like, sure. So they started to do hantavirus work in BSL-3 with the authentic viruses. And of course, the animal challenge studies are BSL-4, at least at RID with these viruses. So they do those as well. And there are some experiments in the paper why are they 4? I guess the concern about large amounts of...
Yeah, it's when you have large amount of virus, and also when you are putting anything in rodents especially, then you have to make sure that you keep everything contained, and the requirement by CDC and NIH is you do them in ABSL4.
All right, well, Jens could probably explain that because he's listening.
Yeah.
So tell us about the screen you did, Rohit.
So just going back to like making viruses with VSVs, so we took the Andes virus glycoproteins. So it really worked for us because these two glycoproteins, GN and GC, that are the only viral proteins on the surface of these viruses that mediate entry. So we could take those two proteins, the gene encoding those two proteins, put it in the VSV, and then make this virus. And for all practical purposes, it now behaves like a hantavirus, at least from the entry perspective.
Right. So because it doesn't have the ability to cause any of the hantavirus disease, you can do this all at BSL-2 levels.
Exactly. And we have gotten rid of the VSV glycoprotein, the native VSV glycoprotein. So, that is not playing any role in entry.
So this should enter the cells the way the hantaviruses do.
Exactly. Yeah. So once we had this virus, then we reached out to Tyne Brummelkamp and Lucas J. So Tyne is co-senior on the paper, and Lucas J., who actually did the screen. So this is the story that it has been covered on TWiV multiple times, the haploid genetic screen, basically where Tyne has the system where he uses these a weird human cell line that is haploid for all the chromosomes. But it's a great tool because what you can do is you can go in with a retrovirus and insert, because ideally you have only one allele of each gene that you are trying to knock out, and you can do this random mutagenic insertion. And Tyne and Lucas are really good at it because, they are able to find things that, a lot of people cannot because of their, expertise. And so when we send them the virus, I was doing for a few weeks, I was doing other things. Then they came back to us with a list of genes that could be potential genes of interest.
So this is a negative screen, right? So
it's a loss of function screen. Yeah. So the way it works is you take this mutagenized library of cells, infect with your virus.
Right.
And the good thing about VSV is once it gets in the cell, it kills them. So it's basically a survival screen. Mm-hmm. So the cells that survive this challenge with the virus are likely surviving because they lack a gene that is involved in entry and infection of these viruses, or at least provides, because it's a survival screen, at least it makes the cells not die.
Actually, when I was teaching, I used to make a huge fuss about the difference between a selection and a screen. Mm-hmm. A screen being you have to look at everything to see whether or not the phenomenon you're interested in is happening. And a selection is where you kill everything you're not interested in. Okay. And the survivors grow. I'd call this a selection.
Yeah, I think we're not being very precise in calling it in this particular embodiment of this, as a screen.
Well, I'm glad. I bet the students are glad you're not teaching anymore.
Yeah. Although, you can use this. Obviously, you can use the same, system to do screens, and
sure,
Brummelkamp has done some really lovely ones with it.
So you look for cells that survive. How do you find out what genes are missing in those?
So because you can track where the retrovirus has inserted, so you can basically use PCR to sequence and figure out the insertion sites, and then you compare those insertion sites and prevalence of those insertion sites in your control library, the cells that you started with, and compare that in the selected population. Mm-hmm. And you're likely to see an overrepresentation of genes that have been disrupted in your surviving populations.
You do this more than once, I guess, right?
Yes. Ideally, nowadays it's become like, you should be doing this 3 times and show 3 replicates.
We used to plaque purify viruses 3 times.
Yeah.
This is, well, that's easier, right? This is a lot of work.
Yeah.
And then you make a heat map of sorts, right? Of hits with the big. I remember you published these with the bigger the circle, the more
right.
time.
So Tyne likes the bubble plots, although they've also been doing fishtail plots.
Fishtail,
because you can also sort of build in a p-value that correlates with the number of sort of the number of independent insertions that you're getting in a specific gene. So I mean, there are different ways you can analyze the data to read out hits.
Yeah. So he actually told you 2 hits that looked.
No, actually. So the funny thing is, the amount of data that you need to put into these stories these days, the most important, interesting question that you're asking, it's actually supplementary figure 1. We have a bubble plot there.
Well, I mean, it's also because when we got the results from the screens that Lucas and Tyne did, there were different sort of types of hits that were of interest. And the one that really jumped out, and that's in the supplementary figure, but it's also because we previously published part of the screen, the results from screen. We pulled out this cholesterol regulatory pathway, the SREBP pathway, this sterol regulatory element binding protein pathway that's involved in cholesterol sensing and kind of interfaces between like sensing and cholesterol uptake and cholesterol biosynthesis. I mean, this is the famous Brown and Goldstein pathway. I always like to joke that if they hadn't done this, we could have used this approach to discover the same pathway, because you pretty much get many of the genes in that pathway. And so we actually did a workup, and a student of mine, Lara Kleinfelter, who just graduated with her PhD, worked with Rohit to characterize these cholesterol hits. And what we found there was that, and Paul Bates in parallel, and Sarah Cherry had also done screens and selections to identify, the same set of genes, more or less, or at least the major ones. And the 2 stories sort of converged, and we both published around the same time to show that these genes, essentially, when you knock them out, you reduce the level of cellular cholesterol, especially cholesterol at the cell surface and in the endocytic pathway. And that seems to reduce infection. Mm-hmm. And then Lara and Rohit showed that you could actually completely reverse this phenotype just by a very brief cholesterol supplementation of the plasma membrane. And it turns out that hantavirus membrane fusion is very sensitive to cholesterol. And that's why we kind of pull these genes out. And so we actually published a paper about this in 2015. But then, all this time in the background, Rohit was working on a single gene in this, from this screen, or the selection, pardon me.
You got it, Rich.
Called protocadherin 1, or PCDH1. And that's what this paper is about, is that single gene, which might have been nothing, but Rohit, it's kind of a labor of love to, really, he kind of worked on this, I won't say single-handedly, but really he was the major mover on, really getting us to the point where we all believed that this was an important. We don't actually use the R word.
You can use
in the paper. Can we use it now? Can we call it a receptor?
Sure, okay. You bet.
So you bet. He moved this work to the point, and it took a number of years because it went in fits and starts, and there were many obstacles, and we can talk about some of those. But there's always this joke about. We actually built a church of protocadherin because we felt like we had to. It's almost like a religious thing, and we had to. There were all these jokes about sacrificing chickens. I don't know. We're both vegetarians, by the way, so we're not actually going to kill anything. But I don't know how luck informs science, and things go in fits and starts, and this is a good example of that. And so actually, even though the screen was done the first 3 months after Rohit joined my lab as a postdoc, we weren't able to publish this until 2018. So that just kind of shows you how long it took. So we actually parceled out the hits from the screen into multiple publications.
So you say in the paper you found one plasma membrane protein in this screen, right?
Right.
Is that unusual?
It is. And if you look at other screens also, usually if you're looking at...
Or selections.
Yeah. What you see is... sometimes you see more than one protein, but these screens, these selections are biased towards entry factors. And so we were surprised, but we were like, okay, it's good that we have only one. If we had 10, we would have to follow up 10, and I'll be here for 60 years.
That wouldn't be so bad.
As long as you're employed, you know.
Yeah.
Right.
Yeah. It also, I think, speaks to doing these screens and selections with the system is really kind of an art that, Brummel Camp is elevated to a very high level. And I think I kind of feel like we were pretty lucky that we did this screen with them because, in retrospect, the biological effect, it's in cell culture tricky to establish with this receptor. And so it's kind of amazing in a way that we even pulled it out of the selection. So we also, and it was just this one little data point sitting right there.
So what was the first experiment that you planned and did with PCDH1?
Well, so around that time, CRISPR-Cas9 technology was become popular. So first, actually, we started playing with talons and trying to make knockouts,
We discovered we had no talent for
no talent for talent.
Good one.
Yeah. So basically, we took a couple of different human cell lines and started with Hapon cell line, knocked out the gene, and then looked at what happens to infection with Andes virus or other old world virus, other new world virus, Sinombre virus, and we also threw in some of the old world viruses in there because we were not really thinking that it's going to be, what we call like a clade specific host factor.
Right.
So when we did that, and obviously we did the complementation experiment where we put back the gene to make sure that, the cells are not, the particular clone that we selected is not messed up because of something, else. And what we saw was very clear that the new world viruses, infection was reduced significantly, whereas the old world virus doesn't seem to care. So,
which begs the question: are you now doing a screen with old world hantaviruses?
We are interested in looking at that.
No comment. No, just kidding.
No comment.
Yes, we would love to, and as I'm sure many other people would, to try and find what the old world receptors are. And at this point, we don't know that there's only one. Or
Sure.
So I assume the glycoproteins comparing old and new world hantaviruses are significantly different. I mean, or
yeah, I mean they have the overall similar topology and fold, but there are there's a significant degree of amino acid sequence divergence.
Okay.
And it doesn't fall so neatly into these new world and old world clades. It's a little bit more messy than that.
Uh-huh.
So in some ways, that sort of geographical distribution is a bit of an oversimplification, but it's useful. Yeah. It's a useful one. Right. But yeah, it is. But it is. I mean, I guess it does make sense in that we are finding with many different, because of the sort of the utility of the VSV system, we don't actually have any of these viruses, right? We are using VSV,
Right.
and we're putting in all these different glycoproteins. And Rohit and other folks in my lab, have really kind of gotten this to work very well. And so now we have a whole bunch of VSVs expressing all these divergent hantavirus glycoproteins, and we are seeing a very clear pattern, which is that every new world virus glycoprotein we've looked at so far requires this protein or uses it, and all the old world virus glycoproteins do not. So there is something that is consistent with this clade description that, affects receptor use, and it, clearly has to be a shared sequence sort of polymorphism. And we are kind of, mapping using that information actually to try and map where the receptor is actually contacting the virus. So we assume it's just that there's a sequence, the surface, there's a sequence difference, the viral surface that creates a different surface that doesn't allow this receptor to bind to the new world virus.
Are there PCDH orthologs that might be involved in old world entry?
Yeah, we don't know.
Mm-hmm.
Do you mean orthologs or homologs?
Other similar proteins that could.
So that's something that we're definitely very interested in, is this notion that
ortholog would be a different species.
Yeah.
Right? So I mean homologs. Yes.
Right.
Sorry. Screen selection.
Yes. Potato. Yeah. I mean, we definitely think that's an interesting idea. I mean, we've talked about doing screens or selections with maybe subsets of these proteins. Cadherin superfamily has lots and lots of genes, so it's
one of the members is the rhino C receptor. Right?
Yeah. And that was exciting because, again, maybe it's, as my postdoc advisor Jim Cunningham used to say, true, unrelated. But, it's cool that you have these two respiratory viruses or viruses that cause respiratory infections in humans that seem to use two different protocadherins. I mean, there's not a lot known about these. They're kind of a neglected group in a way in the cadherin superfamily. So it's kind of interesting that very close to each other, these two respiratory viruses, use two members of this.
So Rohit, at some point you sent these cell lines to someone to do virus infection.
So that's where John Dye and Andrew Herbert step in. So obviously, whatever we find with VSVs, we want to make sure that it matches with authentic virus. So we sent them these knockout cells and complemented cells. And basically, the story is that we see the effect of PCDH1 actually even better with authentic virus than VSVs in almost all cases.
That must have felt good.
Yeah.
Oh, great. Especially because and Rohit found out, to kind of our despair at times that, working with these immortalized cell lines, it's kind of finicky to see a strong effect without the receptor. And, it was really gratifying. And that's figure 1, panel C or panel D, where Rohit actually, showed that this receptor is highly expressed in, endothelial cells, both from human umbilical vein as well as, lung endothelial cells, which are more microvasculature, lung microvasculature endothelial cells more relevant for hantavirus infection in humans. That this protein is highly expressed, way more highly expressed than in these garden variety cell lines that we were using. And also he was able to show very nicely through a population CRISPR experiment that when you knock out this receptor, you see a pretty strong, or at least partially, it's sort of almost like an RNAi experiment where you reduce the levels of this receptor in endothelial cells. You get a much bigger and reproducible effect in these kinds of primary cells than you do in immortalized cell lines. And so a lot of the experiments in the paper were actually done using primary endothelial cells rather than using cell lines wherever possible. Yeah.
So endothelial cells and epithelial cells in the lung are targets of infection?
Yeah, so the major targets the field agrees on are the endothelial cells because you see lots of viral antigen and viral RNA in cells. What we don't know is, so the infection is supposed to happen through respiratory route, but what are the cells that get infected first? Whether it's epithelial cells, how does it get to endothelium? We don't really know. If you look at Syrian hamsters experimentally infected through intranasal route, you can see some evidence of replication of virus happening in epithelial cells initially. But the interesting thing is this protein is also expressed abundantly on airway epithelial cells, which we haven't done those experiments yet with epithelial cells, but that's something we are pursuing.
It's probably how it gets in, right?
We believe so, but we haven't proven that yet.
Because they're exposed, right?
Yeah.
So then you did a series of structure function.
Right.
analysis,
So the typical experiments where we try to narrow down which part of the protein is involved. So this protein has these 7 sort of cadherin extracellular repeats that are structurally very similar to each other, although there are differences in sequences. So we started by, deleting those portions one by one and see which of these domains are important. And, it's sort of makes logical sense. It would be the distalmost part, the part of the protein that's like hanging out there saying hello to virus is the one that is involved in binding and mediating infection.
It doesn't have to be, right? I mean, the chikungunya receptor, the first 2 domains are doing this complicated dance with the glycoprotein on the surface. So, yeah. But, in this case, it looks like it is. So.
Yeah.
Then you made some monoclonal antibodies, right?
Yeah. So before we went to monoclonal antibodies, we could also make express and purify soluble version of the receptor. And what we could we could use that to block infection and show that actually, if you pre-incubate your virus with the soluble receptor before you do infection, you can see block an infection.
And domain 1 is enough to do that.
Domain 1 is enough to do that.
Did you try to select viruses that were resistant?
We have tried that. One of the things is, is that, and, we will come to this, is that PCDH1 is not the sole determinant of entry. Mm-hmm. There are definitely PCDH1 independent mechanisms. Mm-hmm. So when we try to do those selections, I believe what happened was we didn't see any mutations as such, but we haven't followed it up rigorously.
So one thing to point out is that Sinombre is sort of like an Andes mutant when it comes to the soluble receptor, because it binds less well to the receptor and is much less sensitive to the soluble receptor. So those two seem to go hand in hand. So in a way, we didn't do a selection, or we tried to do a selection for escape mutants, but with Andes. But in some ways, Sinombre almost behaves like an Andes mutant in that regard. Mm-hmm. It's not completely resistant, though. Yeah.
Okay.
In fact, by binding with lower affinity to the receptor, it actually seems to be more receptor starved on cells. So that if we always see that when you deplete PCDH1 on cells, that Sinombre gets its butt kicked, pretty hard compared to Andes, presumably because of the lower binding affinity.
It needs a higher concentration because it's got a lower binding affinity.
Exactly, and conversely, it's more resistant to the soluble receptor.
Right.
So just like it's been mentioned on TWiV multiple times, we didn't do that experiment, but nature has already done it.
Yeah, sure.
Yes.
Well, that's what scientists say when they actually can't figure out how to do the experiment.
I think nature's done all the experiments. We just
true.
have to repeat them. All right. What about the monoclonals? What are they doing?
Yeah. So then we were interested in making antibodies against the EC1 domain, and actually we reached out to folks at University of Toronto. That's Jason Moffat, Sastev Sidhu group over there. They use a phase display library to pull out a bunch of monoclonal antibodies that were specific to different portions of the PCDH1 protein. And we had 5 or 6 antibodies that were specific to EC1 domain. And when we tested them, it sort of, agrees with the rest of the data that EC1 specific antibodies block effectively infection with the New World viruses. And just like Kartik was alluding to, Sinombre in this case is more sensitive to the antibody because it requires much more PCDH1.
So far, all these experiments you did and submitted with the original paper, right? None of these have been requested.
Yeah, I think that's pretty much the case. Yes.
Okay.
There might be some little. I think we threw in a few more viruses. Yeah. But whatever. Those are details.
Yeah.
Now there's this section which I thought was very striking. Maybe you could comment on it. We used 4 distinct approaches to evaluate the hypothesis that the glycoproteins directly recognize and engage this PCDH1.
And introducing rigor.
Yes.
And reviewers asked us to show it 3 more ways. So
you had one. You had one, and they asked for 3.
No, we had 4, and
they were asked for
I don't know.
more.
I have to say, maybe we kind of, there were periods where we weren't sure, because there were all these confusing things, doing things in cell culture. The effects were sometimes modest on certain cell lines, and we previously, with a lot of the same people, John Dye, Tyne Brummelkamp, had discovered NPC1 as the receptor for Ebola, which turns out to be, we think, a universal filovirus receptor. And there it's stark. I mean, you knock that protein out, there is no virus infection at all. You knock it out in mice, the mice are going to die of Niemann-Pick disease, but, they don't even seroconvert when you infect them with 10 LD50s of Ebola, so this was not quite as strong because, we always got some infection even when we knocked out the protein. So I think we were even just for ourselves, I think we wanted to really convince ourselves. And then, once you convince yourself and you write the paper, then the reviewers remain unconvinced. So you have to do more things to convince them. Okay. And I think that's fair. I think the paper is better for it.
Yeah, definitely.
Yeah. Also, in fairness, there are many, many examples of this sort of receptor biology thing where people think they have a receptor and it turns out to be wrong.
Yes.
Okay? So this is an area where, in particular, where you have an apparent background of of entry that is not mediated by this particular receptor. This is an area where rigor is really appropriate.
Yeah, I think probably we didn't talk about it this way, but I'm sure this at some subconscious level animated how we went about doing this work, because we wanted to be sure. And, we didn't want to have to, science, I guess, is always something in progress, but you work in progress, but you want to publish a strong paper where you have as few holes as possible. And I think that's
right.
Yeah. And one of the good things about having these collaborative big projects is whatever we do gets validated in 3 different labs anyways.
Yeah. So the reviewers wanted 3 more ways. Did you convince them that was overkill?
Well, so I think when we submitted the paper, we had 2 major assays. And Rohit, you can correct me if I'm wrong. But so actually Megan Slough, who's a graduate student in my lab, worked closely with Rohit on this. She developed this really what I thought was a nice assay where she infected cells with VSV expressing the viral glycoproteins. And then she was able to show that she could decorate only those infected, and this is in the supplement. She was able to decorate only the infected cells with the soluble receptor. So it's sort of like a reverse experiment where instead of having the receptor in the cell, you kind of have it in solution.
So it's figure 7A and B and C.
In the supplement. And so it's nice because she was able to do the controls pretty well. I mean, she was able to show that if you express a VSV, infect the cells with a VSV that express an old world hantavirus glycoprotein, there is no binding. And then you can block this binding, you can block this binding with the antibody against the receptor. Right? I mean, but it's not a purified protein binding experiment. It's still on cells, so there's, I guess, always some room for doubt. So we did another experiment where we put the receptor on the plate, the soluble receptor on the plate, and then added a labeled VSV expressing different glycoproteins, and we were able to show specific capture of the virus by the receptor. It depended. So again, consistent with what all the other things we'd shown. Andes seemed to bind better than Sin Nombre, and the old world viruses didn't bind at all. This interaction could be ablated. This binding ELISA basically you could block binding by using either the soluble receptor or the antibody. So all of this stuff hung together, but I think there were some, none of these experiments are biophysically that rigorous, right? So, so that was one issue, and then there was another issue that I'm blanking on, which.
So another one was. Showing a classical co-IP experiment where you run blots after pulling down, have the receptor.
Like, how do you know there isn't something else on the virus particle that you didn't, that is incorporated somehow that's co-purifying with the glycoprotein, that's actually binding to the receptor and not actually binding to the viral glycoprotein itself. And so I think,
So we did those, and that's again in supplement.
Well, but there's one experiment here where we were able to, so we collaborated with Nicole Tischler in Chile, and for a number of different things here. But one of the key experiments that went in the revision was Nicole was able to send us Andes sera from Andes patients. So these are human serum samples, de-identified. And we were able to show, and I think Megan did these experiments, right? So Megan was able to show that these antisera could specifically block virus receptor binding in the ELISA. And so that suggests, I mean, again, you can always come up with alternative explanations. The simplest explanation was that you have these glycoprotein specific antibodies in the serum, and they're binding to the glycoprotein in our VSV particles and blocking infection.
Right.
Sort of, I mean, I suppose you can't rule out the possibility of protein X, till you have a crystal structure of the glycoprotein.
You can go on and on forever. You can go on and say, well, what about this and this? But at some point you have to publish it, get it out there so other people.
It took us long enough. Right?
I mean,
I really like this one with the human serum. I wrote nice beside it in the journal. So,
So this was, in a way important experiment because so far we were blocking receptor binding using antibodies on the receptor side.
Yeah.
So this was a nice complement experiment.
I have to point out that none of this was in our receptor paper of 1980 whatever, which was among the first virus receptors because we didn't have any of this stuff. We had very minimal, which is good. for us. We didn't have to do all this.
I guess.
But the bar keeps getting higher.
To
and
whom
higher.
much is given, much is expected, or something.
Yes.
So
Yes.
you have a lot of tools. You got to do more things.
Yeah, that's true. It is absolutely true. But I would argue that at a certain point, you stop and get it out there, and then, the field will look at it. And if there really is something hidden that you missed, it'll come out if it's important. On the other hand, if it's not important and nobody works on it.
Yeah, but I think, I don't know. There's also this phenomenon where things get enshrined in the literature. And then, when you go back and look at the primary data, you're like, there's not a whole lot here.
Yeah.
So why is everybody, writing in their papers like as if it's carved in stone?
Oh yeah, that happens, and then you get messed on study sections.
Well, but that also becomes clear because at some point somebody does an experiment, gets a result that doesn't make any sense at all with respect to the previous results, and then they go back and they say, well, this other stuff was kind of weak, so let's check it.
Yeah.
And then it is ultimately self-correcting. But yes, that could be very annoying.
It might just take a long time.
You're talking about my PhD project, which involved basically undoing something that had become enshrined in literature.
Also depends on what lab it comes from, right? The more prominent the lab, the less likely it is to be unenshrined. Phil Sharp used to say, it's better to be first than right.
That's really cynical.
He used to say it all the time. Just get it out there.
Yeah.
I agree. Not with him, with you. It's cynical.
Being cynical.
Well,
yes.
In this case, I hope we're both first and right. And,
yes.
We'll see.
Sure looks pretty good to me.
Yeah.
So let's finally go to the animal experiments.
Right.
Actually, since we were talking about Felix Ray, so this is in figure 3 is the experiment that Felix Ray's lab did, where they actually took purified VLPs, virus-like particles, which only have the glycoprotein, so completely divorced from the VSV system. Just by expressing these proteins as recombinant proteins, you get these particles out. They purified them, and then they put them on a biosensor, and then they flowed the soluble receptor that they produced in their own lab. So this is panel E of figure 3, and they showed binding and its nanomolar apparent affinity.
But you don't know that that's the only glycoprotein in these vesicles, right? There could be other things.
Well, these are actually like purified little particles.
Right, so that's pretty clean.
Yeah, I mean, as clean as a VLP prep, I guess is good. But there is actually a tag on these VLPs that allows them to purify these particles, and they've done EM and run gels, and some of that stuff's in the paper. It does look very clean, but again, to really prove that this is a two-protein interaction, I think you need structural evidence to completely nail it.
Yeah.
So, which we're working on. So, with Felix.
Yeah.
So.
But even then, you could. So you could see, but maybe there's some minor protein that's essential in there that you've missed. But
But then at the other end of it, other end of the spectrum, you did this animal experiment, which is kind of asking the big question: is this relevant in vivo?
Yes.
So did you on purpose say it was a gold standard model, the Syrian golden hamster?
Golden hamster.
Were you being funny?
No. Well, yeah. Well, I love bad puns and these sort of wordplay. Too much to my 14-year-old son's chagrin, but actually pretty much I ran my lab's chagrin. But it's it is pretty much the only model. Mm-hmm. So calling it a gold standard model when you only have one model is kind of
so you
a little disingenuous.
So deer mice are not a model.
No, because you don't get disease and
okay. And this is for pathogenesis. Got it.
And also, genetically engineering deer mice as well.
Well, we
got it.
So there is a non-human primate model for Sinombre, but it's tricky because, and this is something you see with other hantaviruses as well. Once you tissue culture, once you grow these cells in culture, the viruses in culture, they seem to become avirulent. So in order to get a Sinombre that's virulent in non-human primates, you actually need to produce that virus in deer mice. And similarly with Pumalavirus, which is an old world hantavirus in Northern Europe, which its host is called the bank vole. In order to get a Pumalavirus that can make a monkey sick, you need to grow that virus in bank voles. And this has become...
which is very expensive.
Yeah. Well, haha.
Yes.
I totally missed that. Nice one. But this is actually kind of an issue for us for like multiple reasons, because we actually, something I guess, we're doing that's not really in this paper So we are now we're part of a consortium. It's a NIH U19 grant, and we call it Prometheus. And the goal of it is to actually develop a human antibody therapeutic against hantaviruses. That's one of our goals. And even if we have the greatest antibodies, it's going to be quite difficult to figure out how to test them in a relevant model. So the animal model development piece is sort of a big problem with these agents, and that needs to kind of.
So why does the virus have to be passaged in its normal host in order to elicit disease?
We don't know actually for sure.
There's a small protein that's needed for attachment.
I mean, my guess, and there's some evidence for this if you sort of, put it all together, but, there do appear to be genetic changes in the virus that are associated with adaptation. And, those changes presumably cause the virus to become avirulent. But this is a good question. I think there's a lot of work to do there to really.
But in this Syrian golden hamster model, you don't have that problem, right? You can take the virus straight out of cell culture and go into the hamster.
Yes, you can. And it's quite so when you give the virus intramuscular, it's a uniformly lethal model. So it's and it does recapitulate aspects of the.
Yeah, actually, you can in fact. Syrian hamsters with the Andes virus, a bunch of different ways. You can do IP, you can do subcutaneous, you can do IM, intramuscular, you can do intranasal. Pretty much every way, depending on, of course, there are differences in the dose, but you pretty much get like lethality.
Right.
And you get, and the virus goes systemic, but, the animals develop this very severe lung hemorrhagic sort of symptoms, lung pathology. So this is a very useful tool to screen, to look at pathogenesis, but also to screen vaccines and antivirals, and has been used. And hence we call it a gold standard.
Yes.
Model.
So tell us what you did.
So, as you probably gathered, mice is not a great model for this because they don't even get infected. We also tried ourselves, and other people have tried it multiple times. So we were looking for a way to make these knockout hamsters, basically. So we, as far as we know, there's one guy who has published this, who's Zonda Wang, and his postdoc Rong Li, who has been working on genetically modifying, using CRISPR-Cas9 technology to generate knockout Syrian hamsters. So we reached out to him, and we worked together to design guide RNAs and this and that, long story short.
Very long story. So we actually made a knockout. So Bromocamp actually, made a knockout mouse, and we figured we'd make it work. Maybe you don't get good. Maybe the mice don't keel over, but maybe they'll replicate the virus. Maybe we can immunosuppress them. And so, Bromocamp sent us the animals, or at least, right? I'm trying to remember exactly how it went down, but at some point these animals made their way to USAMRIID, where Andy and John, tried to infect them, pretty much did everything but shove a pipette in their eyes.
Actually, those knockout animals didn't go there.
Okay. They were just trying to infect wild type.
They were trying the wild type because the experiment is, a loss of function experiment, right? Sure. So you should be able to infect wild type and then no infection.
But they just couldn't.
Yeah.
Just C57 black.
They tried lots of different things,
including interferon alpha beta receptor knockouts.
You couldn't even see. There was no. They couldn't even see viral replication by qPCR. So really does look like the at least Andy's virus does not.
What you need to do is take the collaborative cross parental strains.
I was just thinking that.
You will find one that gets infected by this virus.
Yeah. So we've been talking to Angie Rasmussen about that. She did that for Ebola. And,
yeah. For sure. Ralph Barrick was here a couple of weeks ago. And you will find, for any virus, some mouse in that original, the set of parental strains that can replicate.
That would be great.
And and then you can map it and so forth. Yeah.
Yeah, that would be great. But, in the meantime, we had a 2-year detour trying to generate these knockout hamsters.
Yeah, which is, a lot of hard work from Rong Li, who's also co-first author on the story. So
I guess you take out eggs and you knock out the gene in them, right? And reimplant them. Is that how it works?
Yes, exactly. So you micro-inject the guide RNA and the Cas9 enzyme so you take the
ova.
Into the ova, and then you implant them, and then you can get the pups, and then you screen them.
Just what the guy did in China with CCR5, right?
Right.
Yeah. Well,
but with hamsters, it's okay.
Right.
You bet.
And
Well, we didn't ask him.
yeah.
Right. Well, true. But
Well, to be clear, for all these experiments, we had all the proper.
I got it. I'm just kidding.
approvals.
so now this is you finally you get this 2-year project just to make these knockout hamsters, and this is one paragraph and mostly supplemental data in the paper, right?
Yeah,
pretty much.
Yeah.
But it's a great experiment, and so you infect these hamsters that lack PCDH1, and what do you see?
Yeah, so what we did is we actually did this experiment just to be sure. We did 2 independent experiments, each one of them having 10 to 11 animals, wild types and knockouts. So with wild types, if we challenge them intranasally with 2,000 plaque forming units, so we did do plaque assays in this paper.
Very good.
Well, we as in the royal.
Yeah. Andy did the Andy's plaque assays, I guess. So when we challenge them, what we see is more than 80% of the wild type animals succumb to disease. Whereas if we challenge the knockout animals, we see more than 80% of them surviving. So what we see is not complete protection, but there is a significant protection.
There's no protein in the knockouts, right? As far as you can
tell. So that's in supplement figure 10.
So I just think it's unfortunate that it all has to be supplementary data, right?
Yeah. So we confirmed, of course, by sequencing and then also by running Western blot to make sure that the protein is gone.
Yeah.
And then we challenge them.
Now, so you could take cells from these hamsters and do the same experiment and find out what other protein is allowing entry, I would guess, right?
Right, or just take other cells.
Yeah.
You know, we have a bunch of knockouts of cell lines.
So is this a universal observation that if you knock out PCDH1 in any cell, that you always get a little bit of entry?
You see significant difference. So depending on the cell type you are looking at, in garden variety cells, you can see 60 to 70% reduction, sometimes 80% reduction in infection. And in primary cells...
Garden variety, I like that.
Garden,
yes. But in primary cells, whenever we do this, we see 95 to 98% block.
Also depends on the virus. So Synombre, as we were talking about before, is more needy for this protein. So if we could, and we can actually do a lethal Synombre challenge in hamsters, because it only works with Andes virus. But presumably, if we could do that, we would find that virus would be well and truly hosed these knockout hamsters. But, that's just a speculation.
It was interesting. This, I guess, a little bit of a side note, but the PCDH1 knockout hamsters seem fine.
Yeah, for the most part. We didn't see any pathology.
Right. Right, but they're living normal lives and, viable. They reproduce.
Oh, if they could talk, Alan.
Yeah, I know. Maybe they're not enjoying the opera as much.
they may go on and on about their health.
And it's interesting because this protein, we talked about it very briefly in the paper, just mentioned it one line, but this protein has come up in multiple GWAS studies as being an important determinant of bronchial hyperresponsiveness, which is sort of a precursor to asthma. Also, atopic dermatitis. So, and there's I'm blanking on the group in Belgium. That's
Gerard Koppelman.
Gerard Koppelman's group has actually done probably the most out of anyone in terms of looking at the cellular function of this protein, and it does seem to be expressed in lung epithelial cells. It seems to be important for maintaining barrier function. Its expression level changes in different situations. So it's doing something, but it doesn't seem to be essential, at least in this context.
Right. Maybe there's something else compensating for it.
Yeah.
Would it be worthwhile to take a HAP knockout and redo the screen?
We actually did that.
We did that. Yeah.
Okay. And you don't want to talk about it?
It was supremely unedifying because what ended up happening was that we didn't see PCDH1. Well, that's good.
Yeah.
But we also, all of the cholesterol hits just got a lot bigger. So we were hoping we would find something else other than PCH1 that could be like a secondary receptor or co-receptor or something, but that didn't pan out. Yeah.
No plasma membrane proteins in that.
Nope.
Turned out. No. Okay.
Actually, there was one.
Oh yeah.
What is it called? CDH.
We got another cadherin, and we were super excited.
CDH4. I was like, this is the other protein because I knocked it out. It didn't do anything. So we don't know why it showed up.
The fact that you can just say that glibly is amazing, right? I knocked it out, and it didn't.
Right.
Yeah. This is one of my pet peeves, actually. I'm simplifying these things, saying like, oh, we did CRISPR screen or we generated CRISPR knockouts. Agreed that, as compared to what it used to be, it has become much easier, but it still takes weeks and months.
Still a lot of work. Yeah.
But we dreamed about this. Yeah. We couldn't do it at all. And then when we started to be able to do it, only certain labs could do it because it was just absurdly circuitous.
And we knocked out a gene in a rodent was a Nature paper. It wasn't a paragraph in a letter.
Right.
That was
that's scientific progress, I guess.
Yeah, absolutely.
All right. That's a good story. Now, you told me they wouldn't let you use the word receptor in this paper. Mm-hmm. Why not? Did they tell you why?
Well, actually, I mean, that's not technically true.
Okay.
When we sent the paper to Science, they rejected it. I mean, they reviewed it. It was a mixed bag. So then we decided we would take another shot and go to Nature. And at that point, we'd been talking to Felix Ray about a structural collaboration, and that was kind of getting underway. And, we also asked for his input, and also Matt Evans at Mount Sinai, who's a friend, and, took a look at the paper. And they both felt that that the word receptor is polarizing,
It's absurd.
And that we should just take it out and let the data speak for itself, or speak for what's
essential for cell entry.
So, we're like, okay, whatever, we'll just not call it a receptor. And so when we sent it to Nature, we just completely removed that word.
I think that's absurd that one word makes a difference. And anyway, this is clearly a receptor in my understanding of what receptors are as well, but there's no universal definition of what a receptor is, right? I could make a universal definition. It's in my textbook.
I think a lot of people may interpret it to mean it's the receptor.
Right.
As soon as you use the word receptor, they're like, oh, this is the receptor.
Right.
Well, it's a receptor. It's essential for cell entry. It's, I mean, they're...
Well, it's not essential either, right? Because it's very important. And, and that's why we wanted to really get that hamster experiment, because, there are so many papers where it's just some in vitro thing. And, I think... what we really wanted to be able to show, hopefully, we didn't know if it was going to work, but that this protein was important for pathogenesis. I mean, so, call it what you want, but if I knock it out of, hamsters, they don't die. I mean,
the disease.
It depends what you mean by essential.
Yes.
Right? So on TWiV, we did the paper of the bat influenza virus receptor, which is MHC molecule, came out in Nature. Right. Shilka Sturz. I met her in Rotterdam, and she said they would not let us use the word receptor in that paper.
Yeah.
For similar reasons, right? Which I think is absurd.
But maybe part of this is because there have been so many false alarms.
You let the literature correct itself. That's the way it goes, right?
True.
I mean, as we talked about before, we called the polio receptor in 1980-something a receptor with far less data. And if no one has yet proven it wrong, and that's the test of time and experiments, right? Yeah. And so, all of this is receptor for sure. Everything you have in here is a virus receptor, I think people are overthinking it.
If it, walks like a duck and quacks like a duck, it's a receptor.
Overthinking can inhibit. I don't think that's a good thing to do.
Yeah.
What did Jim Cunningham used to say? Correct, but...
True, unrelated.
That's a good title, Alan, right?
True, unrelated. Yes.
It used to make me crazy at the time. But in hindsight, I appreciate it a little bit more.
So, Rohit, you're continuing to work on this system, right?
Yeah, so one of the, there are many questions here, like what is actually happening mechanistically. Right. We know that there is a direct interaction between these glycoproteins and this receptor, but we want to map it further.
And you're doing structural collaboration with Felix Ray, right?
Yes, exactly.
Is acidification required for entry of these viruses?
Yes.
Okay.
Yeah.
And we're also really interested in understanding, because there are other putative receptors for these viruses that have been described. I think, not to impugn anybody, these are, in the field, I think, kind of regards them as receptors, but, I don't think they've undergone the same level of scrutiny in vivo, which for me, that's crucial because if you really want to prove that something is a very important for infection, call it what you want, receptor or entry factor or whatever, you need to be able to show that it matters in a relevant, animal model. And for a lot of these molecules, it hasn't really happened yet. And sometimes it's because it's really hard because these are really essential proteins, or you can't generate animals that are viable in the same. We just got lucky with this one. These hamsters could have exploded or something, without PCDH1. So there's that. But, at least in vitro, I think in tissue culture, we really want to see, we want to re-examine like a lot of these other receptors, and not from a skeptical standpoint necessarily, but really to understand if there's crosstalk, what are the situations in which they're important or not important, and is there some kind of I love the model that Charlie Rice and others have kind of developed for HCV entry, where it's so complicated and there's all these different molecules that kind of work together to get virus entry to happen, and is that true for hantaviruses, right? And and what about the old world viruses about which we know nothing? What about the mini pizza batfish hantavirus? Anything about that?
That's what we need is more research on the mini pizza batfish.
Do that, and then you can come back on and talk about it.
That sounds good.
All right.
Yeah, and then actually, what's going on in other hosts, right? Even for these viruses. Humans are not the natural hosts for Andes and Sinombre. So, are the natural hosts of these viruses using the same molecules? Does this receptor matter for host range? These are all questions that like Rohit is very interested in.
So you're going to stick around for a while and do this?
Yeah.
Okay. The dangers posed by chemical and biological threats are constantly evolving, requiring innovative solutions. Could your research help provide the next generation of protection from weapons of mass destruction or a potential pandemic? Submit your abstract and answer the call for papers for the 2019 Chem Bio Defense Science and Technology Conference. Submission is open for both poster and oral presentations across a variety of topics. The conference is hosted by the Defense Threat Reduction Agency and provides the opportunity to collaborate with more than 1,500 of the most influential scientists, program managers, and leaders in the chem biodefense community. For more information and a complete list of topics, visit cbdstconference.com. Submit your abstract by Friday, June 21st, and help shape the future of chemical and biological defense. The website's cbdstconference.com. Submit your abstract today. Now. I always like those ads where they say, buy it now. And that kind of makes me not.
You need like a really good 800 number. Dial 1-800-777-7777.
Operators are standing by.
Yeah, that's right.
Operators are standing by, yes. In a country not close to you. Kartik,
yeah.
I just wanted to ask you to explain this paper. Ebola virus, but not Marburg virus, replicates efficiently and without required adaptation in snake cells. It's just published last November.
Joe DeRisi.
And I like snakes, so I was just wondering what's this? Yeah, Joe DeRisi. Jens Kuhn. Whoa, Jens is on it.
Oh, you didn't see that? I'm like some bit player in that story. You should ask Jens.
Like an all TWiV
And
paper.
Mark Stenglein, who I think of first for snake viruses.
Snake viruses, that's right. Oh, so you're a bit player?
Yeah.
Oh, we don't have to talk about it then. So Ebola replicates efficiently in snake cells. You said, are there snake filoviruses? I guess so. There probably are. But this is actually human Ebola virus, right?
Yeah, nobody's isolated a snake, filovirus yet. That doesn't mean there aren't any. And I think, there's always this interesting idea that, snakes could be part of the life cycle. Because, with Marburg virus, for example, in these caves where bats roost, there are also pythons and, snakes that kind of, they're predators of the bats that fall from the sky.
So the receptor presumably in these snake cells is NPC1, right?
Yeah.
And
so
does this imply that Marburg, there's a post-receptor block for Marburg?
That's what it looks like.
Because there's no adaptation.
Yeah. So our role in this paper was to do experiments with the VSV system, looking at entry.
I see.
And, I think the real effect here is really is post-entry. So what we did find, we published in a different paper. So these are Boyd snakes, right? Like, constrictors. The NPC1 of this particular group of snakes appears permissive for Ebola. But we published one of my students, Esther Ndungo, who graduated a couple of years back. We had a paper in Sphere where we showed that NPC1 from vipers, so different group of snakes, cannot be used by filoviruses for infection for the most part. So there's an amino acid change. It's actually quite amazing because it's a phenylalanine in permissive species, and in these vipers, it's a tyrosine. So it's single hydroxyl group in a 1,000 plus amino acid protein basically prevents infection.
Cool. Yeah.
Because this residue sticks in this hydrophobic pocket in the virus, in the binding site, and putting hydroxyl there basically just completely kills binding. So and I think Jens is quite interested in that work, and we're curious about these other, Boyd snakes. And it turns out that actually constrictors have a completely functional NPC1 for filoviruses, which is interesting.
Cool.
And I guess it's involved in cholesterol transport, right? In snakes.
Yeah. And so one of the amazing things about this protein is that, it's so highly conserved, it's under strong purifying selection to preserve its function, that we could take human NPC1 and replace like a 300 amino acid domain from vipers. Humans and vipers are like so many millions of years apart, right? And you can take a huge
chunk. Depends on the humans.
Yep.
Yeah, that's true. You could take a huge chunk of this protein and make a chimera where part of it is a snake and most of it's human. And that protein is completely functional for cholesterol transport in tissue culture, at least. So we should make a mouse with it and see what happens.
Alan, that means some people are snakes, right?
Yes.
And yet this protein is basically undergoing accelerated evolution. We think because of this coevolutionary relationship with filoviruses or other viruses that use it as a receptor.
All right, so there must be filoviruses of snakes. We just haven't found them.
Maybe. Yeah, I don't know.
We did a paper last time. They found Delta-like viral sequences in boas. Human hepatitis Delta, which was thought to be only human. Now, snakes and birds.
Well, if they have arenaviruses and snakes, why can't they have filovirus? Sounds good to me.
An arena for snakes. That was the name of that episode. I remember that one. Okay, let's do some picks. I hope you guys brought some picks. If not, you can always go to your...
you can toss out a shovel.
Your list book.
Oh, yeah.
You want me to come back to you?
Sure.
Think about it?
Yeah.
Rohit, do you want me to come back to you? Rohit. Do you want me to come back to you, or do you have a pick? Come back. Okay, Alan, what do you have for us?
I have two sites that offer basically the same service at slightly different prices, the TWiV and Trint. And I'm picking these because I think they may be useful to some listeners. They certainly have turned out to be extremely useful to me. But also because I think we ought to look into them for TWiV. These are cheap automated transcription services. So you send them an audio or video file, and you upload it. And let's see. So TWiV is the cheaper one, 10 cents a minute. And Trint is a little bit more than that. They turn around a transcript of your file, and they're fast. I mean, I sent them a 30-minute presentation, some 30-minute presentations for a meeting, and most of them they finished them in like 4 or 5 minutes because it's being done by a computer.
Yep.
And it gives you a transcript, and the quality varies. I do find Trint is maybe a little bit more accurate, especially for people who have accents. But for my purposes, where I'm just using it to work from for my writing, either of them works pretty well because I can figure out what people were saying. And I think for a podcast, it would be perfect because really the main use of this would be for people to find where in the episode they want to listen to something, or for search engines to index the episode. And it gives timestamps about every minute there will be a timestamp. So if you find something that's badly transcribed and you think you want to listen to it, you can just go to that timestamp in the audio.
So you think we could just put this online without editing for our podcast?
I think you could just take this. You could just take one of these transcripts for each episode, and 10 cents a minute. We generally go on for about 120 minutes, so $12 an episode, right?
That's cheap.
Yeah. It's real cheap, and you would end up with a transcript, and it'll just identify people as speaker 1, speaker 2, and as I say, it's not perfect, but it will give you a basic transcript of the episode, which people could then use to find what they want to listen to, or it'll help us show up in search engines. And for
how does it do with scientific words?
Yeah.
It's a real mixed bag. It's I'm doing a conference right now. I'm covering a conference that was all about induced pluripotent stem cells and hematopoietic stem cells, and I'm reading transcripts about Emma's poetic stems and this and
well, and the reason I asked that is because even just the ordinary voice message transcription that we get,
yes.
If you record something. I mean, if somebody leaves a voicemail and then you get an email with the transcription, I mean, some of them are just hilarious.
Yes, they can be very funny. But certainly for my purposes, I can read it and I can say, oh, I know what that says. And for something like a podcast, it's probably good enough for what we want it to accomplish. But if somebody really wants to make the thing accurate, you've got now a draft transcript with timestamps in it.
Mm-hmm.
So you can go through, and both of these sites have editing systems on them that you can either edit it online, or you can just download the text and edit it in your favorite text editor.
I'll give it a try.
And it automatically changes receptor to entry factor.
Right, yes.
Entry factor. Now, no factors. TWiV doesn't do factors unless they're not proteins, nucleic acids, lipids, carbohydrates. What's left? Ions.
Does it convert reverse genetic system to infectious clone?
We'll make sure
I don't think so.
We'll
but
make sure
you might be able to do a search and replace.
We have to have a TWiV customized
right
computer program.
Yeah.
All right, that's cool. Are you guys ready?
Yeah.
Okay, Rohit, what do you got for us?
So this is slightly different from what we talked about, but this is, a lot of people are interested in understanding how antibodies actually work structure-wise, right? Mm-hmm. So this is a recent review article from Andrew Ward's lab. It's about trying to draw some common themes about studying how antibodies basically neutralize and block infections with HIV, influenza, and which is the third one, filoviruses. So this is a pretty good review, I think. And so it's in Nature Microbiology. So yes, it's behind the paywall, but
it's interesting. Yeah, I saw that. Yeah. All right. How about you, Kartik?
Can I plug software? Or does it have to be a paper?
Whatever you want.
Okay, so I wanted to kind of, I guess my pick is Slack. So I don't know if you guys are familiar with Slack.
Yeah.
It's like basically it's like chat for work.
Yep.
And so about a year ago we created an account for the lab, and I think it's become sort of indispensable for lab communication. And one of the things we really like about it is that there are different sort of ways to communicate. So you can create these public channels where people can just chime in and talk about things. You can make announcements. You can, and then there are, of course, private channels for individuals and groups where you can just write to a group one person or a small group of people. And I think the real power is with all the apps that are integrated. So you can connect it to your calendars, like Google Calendar. You can connect it to Google Drive. There are like a million integrations that really allow you to. So in my lab, we use Evernote as a lab notebook, an electronic notebook, and you can integrate that into Slack. So it's really made communication a lot more efficient. I think some of the more senior people I know roll their eyes because they're like, why can't you just meet face to face? But, we still do that. But, this is a really powerful medium when you have, something quick or you have different people or you have an ongoing discussion. And so then more recently, we got this U19 to develop antibody therapeutics, and there are like 10 different groups all over the world that are participating in this. And so I created a Slack workspace for that project. And it's, again, been very useful because it really encourages communication to be open and not get siloed. You have these different projects, and what typically happens in these kind of situations is that, people involved in the first project don't care about what's going on in the second project, and they're having separate calls. And, this way, everything is we try to keep as much transparency as possible in terms of communication, and it's just like, it totally changed how I think about communication. And I'm not necessarily plugging this particular package. I think it's this idea of how you can communicate that takes advantages. There's so many bad things about cell phones and modern communication. Everybody's distracted and looking at their phone, but there's also tremendous value in terms of making communication not just more efficient, readily available, but also more transparent, which I really like, and it totally changed how I think about communication in my group as well as with collaborators.
And it just about has to be better than reply all email chains and conference calls and the other tools that people are currently using.
So my pet peeve is email chains, especially when you have government collaborators. There's 85,000 lines of boilerplate at the end of the email. And this gets endlessly replicated like a virus.
Replicated, yes.
So you have to literally...
It's like a telomere,
Yeah. It's exactly like a telomere. And then, somebody gets left off the chain by accident, and they come in 5 emails in, and they're trying desperately to catch up. And
then everybody forwards the chain to them. Yes.
Yeah. And then somebody else will do reply all on a previous message that doesn't include them. So then we're back to square one. And so
you never know what the latest email is. Yeah. I know that's a big problem. I've thought of using Slack for the podcasts actually, because it get everybody communicating. Because we use a mishmash of emails and so forth. But
you should give it a shot.
I will try.
That's a good idea. Yeah. I'd be interested in that. Yeah,
I'd be onto that. Yeah.
I don't need it for my lab because it's only one person.
And it also allows you many good opportunities. Like when people ask you, "What are you doing?" You can say, "I'm slacking." But you're actually working.
I'm a slacker. Yeah,
I'm a slacker.
Well, in my lab, slack is a verb. So you say, just slack me. Okay,
slack me. Yeah. But it's extensively used in business, right?
Yeah, I think startups and, software companies.
My son uses it in his job. Yeah. Okay, that's a good one. I'm going to try it for all the Twixes. Rich, what do you have for us?
Oh, I have a blog post by an astrophysicist named Brian Koberlein, who has a whole bunch of other posts on his site that all look interesting. This one, his title is From Heaven to Earth. Another tag for this is using Jupiter's moons to determine where on Earth you are. So I have a long-standing interest in what's called the longitude problem. That is the history of determining what your longitude is, in particular for seafaring type navigation purposes. I think one of my early picks years ago was the book Longitude that talked about the solution to this problem, which ultimately involves developing a clock that can maintain accuracy over a long period of time under adverse conditions, such as at sea. However, there was an intermediate, more or less solution, and that is that when Galileo discovered the moons of Jupiter, he realized that the moons of Jupiter could be used to determine one's longitude. And I had heard that frequently and never really thought it through and never really understood it until I finally stumbled across this particular article. And the bottom line is that if you observe the moons of Jupiter, they are orbiting Jupiter, and they have very regular orbits. And actually, the way to time them is to observe when any particular moon eclipses behind the planet, when it disappears. And it's so regular that it is essentially a celestial clock. And you can make tables of what times, according to the calendar, any given moon is going to disappear behind Jupiter. And the important bit is that no matter where you are on Earth, since Jupiter and its moons are so far away, if you have two observers of this event, they will observe the same event simultaneously. So knowing that, if you know what your local time is, which you can tell because you can always figure out what noon is with a sundial, and you can observe a moon disappearing, you know when that moon disappears that it's a certain time in Greenwich, and you know what your local time is, and from the difference between those two, you can figure out your longitude. I never understood that, but now I do. And that's, for me, very cool. It turned out that Galileo's tables weren't quite accurate enough to be all that useful, but later Cassini came along and produced a very accurate set of tables that were used to determine distances and longitudes all over Earth, and it completely changed the map of Europe. Okay, because they got all the distances down right.
All the previous distances were based on the pace length of a Roman soldier.
That's right. That's exactly right.
Which accumulates errors over a few thousand kilometers. Yeah.
And the only other noteworthy thing is that as useful as this might seem to be, it turns out really it's useless for ocean navigation because the boat's rocking all the time and you can't get a good shot at the Jupiter's moons. Okay?
Huh.
But it's good on land.
So it's too close, I guess, right? Because the stars are far enough away that it's...
No, the problem with a boat is you can't make the observations because the boat's moving. Okay.
Interesting stuff. I don't care where I am.
Well, just pull out your phone. Right?
But I can see where some people would want to know. Kathy, what do you have for us?
I picked something which is maybe only marginally scientific, but...
Oh, no, I think this is totally scientific.
Okay, good. Then that's all I need to say. Rich says it's scientific. It's Architectural Digest has identified what they think are the top architectural designs in all 50 states, plus Washington, D.C. And so it runs the gamut from something built in 1800, the U.S. Capitol, all the way to something built in 2018, the National Veterans Memorial and Museum in Columbus, Ohio, and all kinds of really cool things in between. So it's just a really fun gallery to look at and make note of which ones you've already seen and which ones you might be able to see in the future.
Cool. So the Verizon building doesn't make the list, I'm assuming.
I don't know.
No,
what state?
I didn't see it. I looked through all of these.
That's like, I'm just trying to make a joke because that's like that ugly building you see when you're in Brooklyn looking across the Brooklyn Bridge. So.
I like the glass house. It's really
so do I. I was going to
cool.
say it's very
so cool.
Yeah,
I would live in a glass house, but I wouldn't throw stones. Some of these are just amazing. But
would you stow thrones?
What's that?
Would you stow thrones?
Sorry, Alan. What were you going to say? Montana. Oh, that's nice.
I've seen that.
Yeah,
yeah. It's pretty cool.
Some of these are really far out.
Yeah,
you got the crazy buildings in Cambridge.
Oh, look at this, Centra Metro Park. The hell is this? It's in Iselin, New Jersey, which is where I get the train. I've never seen that.
See, that's what I said. You have it now. You can make a list of things that you need to see. Grand
Central Station. That's a cool building. If you haven't been in there, that's very cool. Neat, good stuff. It doesn't have to be really sciencey, but this. I mean, architecture is a science for sure.
Yeah.
Right.
Okay.
No problem. I have a pick, which is an episode of a podcast called The Cyberwire. This is a daily 30-minute podcast on cybersecurity, most of which I don't understand, but I like listening to it. Like people say about TWiV, they listen enough, they start to understand things. But this one is about such a cool story. It's called The Ghost and the Mole, Eric O'Neill's Gray Day. So here's the story here. Eric O'Neill is a former FBI counterintelligence and counterterrorism operative. In the 1900s, he was charged with helping expose Robert Hansen, who was the FBI's most famous and notorious mole. Okay, so Hansen was the worst spy in U.S. history.
Yes.
He, over 22 years, he worked within the FBI as a mole for the Russians. He never got caught. And finally, I mean, he gave away so much stuff. He gave away agents who lost their lives. He gave away secrets. The U.S. had built a tunnel under the Russian embassy in D.C., and he gave it away, so they were giving us false information. Amazing. He never got caught. But at the end, they suspected him. So they charged this young guy, Eric O'Neill. He was a pretty young agent, to go work for Hansen in an office, just the two of them, and somehow get information to convict him. And this is he talks about it in this podcast. He's also written a book called Gray Day, which I've got, but I haven't read yet. It's just amazing. And just to hear him talk about it is amazing, because he said, oh my gosh, I didn't know anything. And here's this guy for 22 years. He's a mole, and we're trying to charge him. So how he did it in the end is very cool. And
no spoilers, right?
I'm not going to tell you, but it's chilling. It really is chilling. It was amazing. And it worked. You know, they caught the guy and they convicted him and blah, blah, blah. So it's a pretty cool story. The podcast itself is neat. But it's a lot of stuff, as I said, I don't get. They use all these code names for exploits and stuff. I don't know what they mean. So, but I like listening to it. All right, we don't. There we go. That is TWiV 549. You can find it at microbe.tv/TWiV if you want to see the show notes and all the links that we give you, the pics and so forth. But you will listen on your phone or tablet in an app, and we would like you to subscribe so that we know how many people are listening. Now, it's even more than ever important to subscribe, because this doesn't affect us all that much, but advertisers are now getting very stringent about counting downloads. And I won't go into that now, maybe another time, but please subscribe if you don't. We could really use it. And, of course, questions and comments, go to twiv@microbe.tv. Our guests today from Albert Einstein College of Medicine, Rohit Jangra. Thank you for coming.
Thank you so much for having us.
Did you like your first TWiV?
Yeah, it's great.
Is it the same as listening?
It's different.
Different, yeah.
But it's way cooler.
It is way cooler. We have a lot of fun. Kartik, Chandran, thank you for coming back.
Thanks for having me on.
Always nice to have you. We like your puns.
Maybe the only one.
Kathy Spindler's at the University of Michigan over in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at turbidplaque.com. He's Alan Dove on Twitter. Thanks, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASV and ASM for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.